afternoon, everyone, and welcome to a new session of this liver seminars edition. I am Melissa Lozano from the University of Salamanca, and it's a great honor for me to participate in this seminar series. I would like to thank the organizers, Dr. Rafael Bañares and Dr. Jordi Gracia Sancho, for giving me the opportunity to host this seminar, which is entitled Building a Bile Duct, Using Developmental Biology to Understand Adult Biliary Disease and Cancer. It is my pleasure to introduce you today's speaker, Dr. Luke Bolter from the University of Edinburgh. For who doesn't know him, Dr. Bolter graduated in cell biology at the University of York. Then Luke undertook his PhD in Edinburgh with Stuart Forbes, where he began to define the molecular signals that direct fate choice in adult liver progenitor cells and understand the molecular events that give rise to cholangiocarcinoma. Luke moved to the MRC Human Genetic Unit in 2014 to establish his own group, the Tissue Growth and Cancer Lab, which works on understanding the molecular interface between health, disease, and cancer. The current focus of the Dr. Bolter Lab is to understand how the signals that govern normal liver development in the embryo are used reiteratively during regeneration and cancer to support tissue repair and carcinogenesis, with the ultimate aim of modulating these signals to limit disease progression. Before starting with Dr. Bolter's presentation, I would like to remind you that we will have time for discussion after his talk. So please don't hesitate to write your question or your comments down on the chat or to raise your hand later to ask by yourself. So thank you very much, Luke, for accepting our invitation and for being with us today. And I give you the word.
I normally talk about the work we do in liver cancer, so cholangiocarcinoma, and some people in the audience may have heard me speak about that before, but I thought what I would do today is talk about some of the other work my lab does. So we are interested in this structure here, and this is the bile duct. So it's a series of tubes. So we work on this set of tubes called the bile ducts, and obviously you'll all know that those are essential for normal liver function, and there are a number of both developmental and childhood diseases associated with dysfunction of the bile duct, but also a number of adult diseases, including cancer. And what we are really trying to do is understand this structure and how this forms, and as Lisa said, to kind of understand this molecular interface between what is normal tissue, what is disease, and what is cancer. And I very much see that cancer is, at least cancer of the bile duct, is a state in which normal tissue growth and repair goes wrong. But I'm going to talk to you about these first 2 points today. How does a complex biliary network form? So how do we go from a relatively simple epithelium into a more complex, highly ordered structure, such as the bile duct? And also, how do those bile ducts maintain their structure in adult life? So why don't they fall apart? Why do they maintain their normal dimensions? So in order to understand what I'm going to talk to you about, I just thought I'd revisit some of the kind of fundamental processes that happen during liver development, particularly bile duct development. So the bile ducts form from a relatively simple epithelial sheet. So in development, all of the epithelia comes from a bipotent progenitor cell called a hepatoblast. And hepatoblasts that are close to the portal vein are specified through a number of signals, actually, to form a biliary fate. So those aren't necessarily cholangiocytes at that point. They're not structured, they're not in these ductular networks, but they are not hepatocellular either. They've separated from that lineage. And then what we know is that after specification, so once we have this ductal plate, that ductal plate undergoes a series of complex morphological movements in order to go from a sheet of cells into a tubular structure. That tubular structure then itself has to undergo subsequent morphogenesis in order to become highly branched. And we really don't know much about those processes. I'm just showing on the slide here. Hopefully I can just get my pointer up. There we go. So on the right-hand side, what this process looks like. So early in development, this is a mouse liver now. You can see that there are cells that are SOX9 positive being specified. So these yellow here, yellow cells are SOX9 positive cells. You start to see these emerge, and then they form these plates, these sheets of cells, which then undergo wrapping and structural rearrangement and morphogenesis. So this really is a stepwise process throughout late development. And actually, we do know a fair amount about how the ductal plate is specified from human genetics. So particularly Alagille syndrome, where patients are born with a very, very poorly arborized biliary tree. Those patients have a loss of function mutation in a Notch ligand called Jagged1, or sometimes have a hypermorphic Notch2 receptor, and Notch2 and Jagged1 interact. So we know that from human genetics, at least, the Jagged1 Notch2 signaling pathway is important for specification of this biliary lineage. And that has been iteratively shown in model organisms, particularly in the mouse and in the zebrafish. We also know from those studies and from other works as well that actually TGF-beta is important in that process. But what we don't know is kind of what happens next. So we are happy that we can get really to this stage where we have an epithelium that is cholangiocytic or biliary, and that is different from hepatocytes. And we know we can get to this stage where we have a very, very highly branched biliary tree, but we don't really know in humans or in mammals generally what happens between these two states. So how do we go from shorter stretches of a discontinuous duct into a complex structure? How do these ductular structures intercalate and form one long tree? And also, how do those ducts then form new branches and finer branches, ultimately connecting the canaliculi into the bark? So when ducts form, they form in order to serve a purpose, and that is to drain bile, obviously. But to do that, they have to conform to some fairly stringent physical rules. So in order for flow to be normal through a duct, they have to be the right width. They also need to be the right length, because there's no point in making ducts that are too short, because they won't serve their purpose. Also, you don't need to commit lots and lots of cellular resources to making too many ducts and making them too long. And also, as they branch, those branches need to be distributed normally. So instead of being hyper-branched, they need to have normally distributed ducts along the length of the main duct. And so what we are trying to understand from a very fundamental level is how you pattern those physical parameters, so width, length, and distribution of branches. But because we don't really know what happens after linear specification in the liver, this has kind of been very difficult. So as I've already said, we're happy that Notch and TGF-beta can specify the biliary tree, but what factors regulate bile duct morphogenesis? And we've started to take advantage of some of the single cell studies that have been published to address this. And really we focus largely on this in this talk. The data I'll show comes pretty much exclusively from this paper, which was a very early publication in the kind of single cell liver space. And what they did was they took a developmental time course from the mouse between embryonic day 10.5 and 17.5. So mice are born after 18.5 days. So these are very, very late embryos. And they isolated livers, and the authors sorted out epithelial cells by using FACS for DLK1 and EpCAM. And then they subject those to single cell RNA sequencing, and essentially could put together a map of the epithelial lineages over developmental time in the liver. And we rationalize that if we can use that data set and define markers that represent terminally differentiated biliary epithelial cells, so those very, very late in development cells, we'll be able to partition those cells out and use those cells to identify the signals and the regulatory factors that allow for the formation of tubes and ducts and really allow this simple epithelial sheet to undergo morphogenesis and become a complex ductular network. So these are now data generated from my lab using that data set. And what we were able to show was that, or what we found very similar to that paper, was that within that data set, there are divergent epithelial lineages. So TBX3 is a hepatoblast marker in this context. And you can see that early in development, so here in these kind of reddish colored dots, there's quite high expression. So the size of the circle represents level of expression. And as you go through developmental time into the hepatocyte lineage and into the ductular lineage, the expression of TBX3 decreases. So that's not particularly surprising. As you differentiate, you lose progenitor traits. And similarly, you acquire hepatocyte traits in the hepatocellular lineage. So this is now RNA expression of CPS1 and SOX9. So this ductular transcription factor. And so knowing that we could happily segregate the different epithelial lineages from this data set, we moved forward to ask whether we can start to identify new biology using single cell. And what I want to draw your attention to is this kind of schematic where in this pink at the end, I'm showing you what I think are the terminally differentiated epithelium. So this isn't real data, but where in the next couple of graphs, you see spots at the end here, we think those are the very, very most mature cholangiocytes in these embryonic livers. And the reason we think that is because keratin 19 is a marker of terminal biliary differentiation. So it's expressed very late, just before the mouse is born, or indeed the person is born. And you can see here that keratin 19 is most highly expressed in these cells that are right at this tip of this kind of domain. So we think these are our very mature cholangiocytes. And when we take those livers and do whole mount immunofluorescence, we can see that keratin 19 does indeed mark these very, very mature, complex bile ducts. We've also stained C31 blood vessels just to give you some orientation. But that's not new. We knew already that keratin 19 is a marker of the ductular epithelium. But using that, we could start to identify the important regulators of tissue morphogenesis. And what we did is we took that single cell data, and you can represent this in lots of different ways. So the UMaps that I showed you, or essentially in these kind of heat maps, where if you look across developmental time, you can look in the hepatoblast to biliary lineage or the hepatoblast to hepatocyte lineage. And then you can interrogate specific genes. So instead of looking at whole populations of genes, you're looking at single transcriptional readouts. And what we were able to show was that in the hepatoblast to bile duct lineage, a number of genes that are associated with planar cell polarity signaling are upregulated in the hepatoblast to biliary lineage. But actually, when hepatoblasts become hepatocytes, those genes are, if anything, suppressed, but just not really expressed at all. And we think this is quite important because planar cell polarity is part of a non-canonical Wnt family that regulates both the directionality of a cell, but also how cells collectively move and migrate. So you can imagine a situation in late development where you are a short stretch of bile duct and you need to elongate and stretch in order to find your next bit of bile duct that you're going to join up to. And so we hypothesize that this planar cell polarity pathway is important in allowing these cells to assume directionality and for the structure of the bile duct to elongate and grow. What I would just highlight from this is that there are major components of this pathway. So Van Gogh type 2, Van Gogh type 1 and Van Gogh type 2 actually, but in our case, Van Gogh type 2 is a major scaffold component that many other proteins center around in this pathway. And the pathway ultimately acts through Rho and other downstream intermediates to rearrange the cytoskeleton. And that'll become important later. But I'll just draw your attention back to here, because what we did was we wanted to try and demonstrate or investigate whether Van Gogh type 2 as a kind of core regulator of this pathway was important for biliary development. And so we are kind of a very mouse transgenics lab heavy group. So we made a mouse in which we fused green fluorescence protein to Van Gogh type 2 in vivo. And that allowed us to look where Van Gogh type 2 and these planar cell polarity complexes are localized to in mammalian tissue. And what we found was that here in fluorescent staining, that Van Gogh type 2 almost exclusively localizes to developing ducts, and these are keratin 19 positive. So these terminally differentiated epithelial cells express Van Gogh type 2, and what we could show was that Van Gogh type 2 actually, and I won't go into the data too much, is very heavily partitioned and localized at the cellular level, and is found essentially on the apical surface of the cell and in the lateral membranes. So Van Gogh type 2 and by extension planar cell polarity is polarized. And we know from fly right through to humans that planar cell polarity is important in this kind of cellular positioning and directionality. But what we don't really know is how planar cell polarity and those complexes kind of translate that into tissue mechanics. So planar cell polarity is a chemical signal that allows for elongation, but how? And we've not really kind of understood that in the field. So we've tried to address this kind of using the bile duct as a model. And what we wanted to determine was whether Van Gogh type 2 interacts with anything that could regulate the structural growth of the bile duct. So we've already specified these cells. We just want to make sure that that they're patterning normally. And so we made use of our Van Gogh type 2 EGFP knock-in mouse by removing the livers and lysing the cellular components. And this was done from E18.5 livers, so just before the mouse is born, where we think we've got kind of morphogenesis of the biliary tree happening. And after lysis, we can capture the EGFP with pull down, and the idea here is that we can do co-IP mass spec. So we'll be able to pull down the EGFP and therefore Van Gogh type 2, and any proteins that are interacting with Van Gogh type 2 will also be captured, and we'll be able to identify those in a really non-biased way. And this is some work we did with Alex von Krietsheim at the Cancer Centre in Edinburgh. So when we look at the results of this, having captured Van Gogh type 2, we were very reassured to see that our biggest, our most abundant protein after cool down was indeed Van Gogh type 2, so our assay works. And I'm not going to lie, this was quite a surprise given it's from embryonic livers. We were pretty chuffed that this was a success. And along with this, we found a number of proteins that are associated with cell-cell junctions. And so particularly things like DSG1 alpha, RAC, which is known to reinforce cell-cell contacts, and a number of others. And when we take these enriched proteins and put them through GoTerm analysis, what we find is that there is an enrichment in our Van Gogh type 2 EGFP pull down for things associated with the desmosome, with cell-cell contacts, with cell-cell junctions. And so what we are hypothesizing is that planar cell polarity physically locates proteins that are required to for cells to interact, but also for those cells to then confer mechanical tension along the length of a duct in a non-cell autonomous way. And if that hypothesis is correct, then if we disrupt Van Gogh-like 2 and therefore planar cell polarity, we should stop ducts being able to elongate, and those ducts will kind of still be made, but they won't have intercalated and formed a branched biliary tree. And so to explore that further, we've used a mouse called Looptail. So this mouse has a mutation in the C-terminus of Van Gogh-like 2, and as a heterozygous mutant, it's viable, it behaves fairly normally, it does have a looped tail, hence the name, and has some stereociliary defects in the ear, but other than that, it has very few phenotypes. Whereas the homozygous mouse is lethal perinatally. It has a number of morphological defects, particularly cranioechistosis, so it doesn't close its neural tube, but also fails to form branched structures in other tissues, such as the lung and the kidney. And so we thought this would be quite a powerful tool to understand whether planar cell polarity regulates mechanical structural maturation of the biliary tree. And I obviously wouldn't be telling you this unless that was the case. So what we were able to show was that in the mutant here compared to the wild type, loss of planar cell polarity affects the formation of tight junctions. So you can see ZO1 here nicely forms in, or the ZO1 tight junctions nicely form in the wild type. But in the mutant, those ZO1 junctions are much smaller, and when we quantify that, that's the case. Similarly, when we look at E-cadherin as a marker of the adherenous junctions, we can see nice, regularly spaced, mature adherenous junctions in the wild type duct, but in the mutant, those junctions either don't form or they're very deregulated. So the E-cadherin doesn't form nice regular patterns. It forms a very kind of discontinuous network. And what we subsequently became interested in is, well, if we know we're disrupting the essentially the ways that cells communicate within this developing duct, can we understand how that's changing the ability of the duct to perceive mechanical tension? And so what you can see here is that in a wild type mouse embryo, so this is again E18.5, phosphomycin light chain 2, which is responsible for conferring tension on the cytoskeleton, is very regularly distributed on the apical surface of these cells. So here you see another developing duct in here. Whereas when we look in the mutant, phosphomycin light chain 2 is almost always lost, as in these two examples at the top, or it's very broadly distributed and no longer has its normal apicobasal polarization. So the phosphomycin light chain is all the way through the cytoplasm. It's in the lateral membranes of the cells. It's all over the place. And we can quantify this using fluorescence intensity. And I won't really go into that, but what we can conclude is that in a normal situation, mechanical tension in a cell is regulated in this kind of predominantly apicobasal way, but actually in a cell where Van Gogh-like 2 is mutated, the phosphomycin light chain is either wrongly distributed or lost. So what is the consequence then of changing this this tissue mechanic on duct formation? And it's that you can no longer form a correctly patterned biliary tree. So here you can see on the left the number of ducts in these livers, and so you've got wild type here in blue, and in this pink color the mutant, and there are many fewer ducts that form. So this is normalized to liver area, and the liver size is normal. So these mice don't just have smaller livers; they are standard size. So it seems that the hepatocytes and the parenchyma and the vasculature all develops fairly normally, but what fails to form is this complex biliary network. So, as I mentioned earlier on, we do think that the bile ducts will form, it's just that they fail to intercalate and mature. And that's what we see when we look at whole mount imaging. So, compared to this very continuous and complex biliary tree in the wild type, when we lose planar cell polarity and tissue tension within the duct, we form a biliary tree that doesn't connect. So you can see these white arrows demarcate places where small stretches of duct have failed to integrate. And the consequence of this, we think, is that if you can't form a terminally structured biliary tree, then your epithelium doesn't differentiate terminally either. And so we lose a lot of keratin 19 expression in our mutant cells, and you can see when we've quantified this, it's reduced. And also these cells fail to present a primary cilium into the lumen of the bile duct. And so whilst they continue to form ductal plate and the progenitors of the biliary epithelium, we actually don't think they form mature ducts. And I can talk a bit more at length, perhaps in the discussion about that, because we've got a bit more data suggesting that that's the case. So what I've shown you there is that in late development, bile ducts elongate by integrating both planar cell polarity to give them chemical directionality and also cell-cell junctions in order to give tissue mechanics and mechanical changes that are required for ducts to elongate and integrate. But if we know that planar cell polarity can affect duct length, how is width managed? So I suppose kind of duct function is a literally a function of the width compared to length. And so width is important. And we know again from human genetics that there are a number of liver pathologies where ducts fail to maintain their normal width architecture. So things like von Meerberg complexes and polycystic disease have these hugely dilated ducts. So we want to know what we can learn really from human disease in order to kind of understand the fundamental principle of tissue patterning. And actually, there are a large number of patient-led, or I suppose kind of patient diseases, which can result in polycystic liver disease, so where the duct becomes cystic. And this includes both autosomal dominant disease, but also a number of isolated polycystic liver diseases or autosomal recessive diseases as well. And there are a number of syndromes and conditions where liver involvement is probably massively underestimated. So I'm going to talk very briefly about the primary cilia and then go into some of our data regarding this. And there are multiple hypotheses of how ciliary dysfunction affects cellular function, particularly in the biliary tree. So there's an idea that the cilia is an osmosensor, that actually chemosensation of bile acids in particular are sensed by the cilium, and that's important for normal kind of ductular homeostasis, or that actually the primary cilia senses mechanical flow from the bile and that it regulates calcium signaling that way. And actually a number of the mutations that I showed you on the previous slide could probably affect different processes within this kind of schematic. But actually what we don't know is, are all of those mutations common, as in regardless of mutation, are the processes that a cyst forms through normal, or duct widening happens the same? So we've established a model where we can delete an essential cilia gene, WDR35, in the biliary epithelium, and in those animals, the cells fail to form cilia. So you can see here in our wild type situation, you have a duct here in pink, and you have these very small IFT88 and RL13B positive cilia poking into the lumen of the duct. When we delete WDR35, we lose those IFTHA RR13B positive organelles, and we form these cysts. And what we know from a lot of work done from various other labs, cilia labs in particular, is that lots of this WDR35 protein, which is part of the IFTA complex, means that the cilia can't mature. So things fail to get loaded into the cilia, and failure to load cargo into the cilia results in its kind of collapse, essentially. And when we do this in the bile duct, what we find is that the bile ducts get wider, so they are obviously cystic, so they get bigger. And we measured this in a number of ways. Importantly, alkaline phosphatase also increases. So there is a disruption of normal biliary function in these mice. I should say this data is now on bioRxiv if anybody wants to go and have a look at the paper and have a really deep read of it. So to try and understand what the cellular processes were that are activated when you transition from a normal duct into one of these widened cystic epithelial ducts, we isolated cholangiocytes from normal mice or mice that have been allowed to develop polycystic liver disease over 12 months. And we used single cell RNA sequencing to try and understand what was changed. So you can see when we cluster the biliary epithelial cells, they cluster quite nicely depending on whether they're wild type, so here in cyan, or mutant, and so this is a WDR35 mutant. And this has allowed us to look at the differential gene expression between these two kind of cell states. And perhaps unsurprisingly, TGF-beta signaling is very highly upregulated, so you can see that within the cystic cells, we have high levels of TGF-beta ligand, TGF-beta receptor 2, and also the downstream effector SMAD3. Nicely, there are a number of GO terms, so gene ontology terms, that are enriched in cystic cells compared to the wild type, which are to do with changes in the actin, so cytoskeletal remodeling, cell matrix interactions, and we'll go on to that in a moment. So all of these data kind of pointed to the idea that there's active remodeling both of the extracellular space around the developing cyst, but also within the epithelium as it gets wider. And we confirmed a lot of these transcriptional changes with proteins. So we can see that TGF-beta receptor 2 is expressed on the surface of the cystic epithelium, but not in the wild type, and that the downstream effects of TGF-beta signaling are indeed activated and translocated into the nucleus. But a 12-month model in vivo is quite difficult to continually justify, particularly when your PhD students only got 3 years to do their project. So we tried to develop an in vitro system where we can model this duct to cyst transition. And so what we've worked up is a method in which we can isolate duct epithelium, so but not as single cells, rather as ducts. And we put those into a basement membrane. And what happens over time is that those ducts start to form cysts. This is essentially the first step of the organoid isolation protocol that's been published a number of years ago. And we can then interrogate this system with small molecules. And we use CYS3, which is an inhibitor of SMAD3, so therefore downstream TGF-beta signaling. And what we showed was that when we treat those duct to cyst cultures with a vehicle or some old CYS3, we reduce the amount of cystic area that forms. So we think that TGF-beta is activated, but also inhibition of that pathway is sufficient to limit the transformation of healthy ducts into cystic tissue. So then we went in vivo, kind of having a bit more confidence to say, well, let's try this. And what we found was that actually, when we inhibit TGF-beta in vivo, so we have cyst-bearing mice, we either treat them with a vehicle or CYS3, we can reduce the amount of cysts that forms in vivo. So those cysts are significantly smaller. We don't actually change the number of cysts, which is interesting, I think. But we do limit their growth. And importantly, we change we reduce alkaline phosphatase in the serum. So we are in some ways restoring biliary function, though we definitely don't go back to what the biliary function of a healthy liver. We also think that this phenotype is driven largely by the production of fibronectin, and I won't really go into that because it just adds another kind of complexity to the story. But we think the remodeling of the extracellular microenvironment is particularly important for the formation of cysts. So to kind of conclude that bit, what we think happens is that when you lose the primary cilia through some kind of genetic insult, be this in autosomal dominant disease, where it's PKD1 and 2, or in autosomal recessive diseases, you activate TGF-beta signaling. We're not entirely sure how, but we think that's what happened. And as you transition into a cystic epithelium, you produce a procystic extracellular matrix, which is very high in fibronectin, and we've been able to show that inhibition of TGF-beta limits this, both in vivo and in vitro. I will say at this point that we have some nice human data that I've got included that shows that actually, regardless of what the causative genetics are of your polycystic liver disease, you essentially activate very similar processes across all genotypes. So this is a cellular response to ciliar loss, not a genotype specific event. So whilst we're happy that we can form this procystic niche, we wanted to then try and figure out, well, what is on the epithelium that is receiving this new microenvironment and changing the kind of structure of the epithelial cells. And so the nice thing with single cell data is you can do some pretty clever computational stuff. And here we do receptor ligand interaction modeling, where we asked what receptors and ligands are likely interacting between cells in wild type biliary epithelial cells, and then how does that change when we compute this with our cystic mutant cells. And what you can see is that in the mutant cells, they establish this kind of new cystic integrin profile. So the number of interactions with beta-1, for example, massively increases compared to the wild type. Again, the number of interactions with fibronectin increases, and we start to see the expression of some novel cyst-specific integrins. The integrin alpha-2, for example, which we know forms a heterodimer with beta-1, and integrin alpha-B is also there. And we've really focused on integrin alpha-2 beta-1 because it seems to be selectively expressed by cystic epithelial cells. And you can see here just some UMAPs. I'll remind you that the cystic cells are down in this region at the bottom, where only cystic cells on the whole express integrin alpha-2. Everything expresses beta-1 because it's ubiquitously expressed protein, but only cystic cells express integrin alpha-2. And when we look at the protein, this is absolutely the case. So you can see here, there is pretty much no integrin alpha-2 expression in the normal biliary epithelium, but when you are polycystic, that integrin alpha-2 is expressed throughout. Nicely as well, in our mutant mice, there's high levels of downstream effectors of integrin signaling, so things like ILK and Rho and FermT1. And because we're interested in tissue mechanics and how tissue mechanics changes, initially in development, but now in polycystic disease in the adult, we wanted to understand whether changes in integrin are associated with remodeling of the cytoskeleton. And using a very similar approach to what I showed you in development, we stained for phosphomycin light chain. And you can see that actually in the resting duct, there is very little phosphomycin light chain at all, because those ducts are static and they're not being remodeled. However, in the polycystic context, there's a lot of phosphomycin light chain expressed because those cells are having to remodel their actin and their cytoskeleton. And this changes the structure of those cells. So instead of them sitting there with nice columnar epithelium, those cells get wider and they stretch and they form this classically cystic phenotype. But I think we've always assumed that those cells, those epithelial cells that form a cyst, are stretched because of luminal pressure, and I don't think this is the case. I think those cells are actively remodeled in order to give more space to the lumen, and they actually get wider and wider. So if that's the case, and we think that integrin beta-1 alpha-2 signaling is responsible for enabling this kind of cyst stretching, the simple experiment, of course, is to inhibit alpha-2 beta-1 in those cysts, and we should be able to reduce the cystic growth. But we tried this, and unfortunately, that's not what happened. So when we gave a highly selective chemical inhibitor of alpha-2 beta-1 to mice in vivo, we saw no appreciable difference in cyst size when we looked at the vehicle versus the inhibitor. And this then kind of took us back to thinking, well, maybe we've oversimplified this. Maybe actually this isn't all about growth. When we look at things like von Meyenberg complexes, those cysts form adjacent to each other. So it's unusual in polycystic disease, and almost by definition, with the poly bit of polycystic disease, you have multiple cysts and multiple structures. So perhaps the extracellular microenvironment and integrin signaling isn't directly regulating growth, but maybe it's regulating some of this kind of polycystic context. So what we did, and this is a bit of a wild experiment, if I'm really honest, is we used our polycystic mouse, so a mouse where we can delete WDR35 in vivo, and crossed it with this this reporter where essentially when you remove a stop codon, you can get a variety of different fluorescent colors expressed. So you can have GFP, you could have YFP, you could have RFP, you can have CFP, and those are stochastic. So in any cell, you don't know whether you're going to get one of these colors. You can also remove the whole set and get nothing. But the idea here is that we will be able to assess in our cysts whether we got polyclonal cysts. So do cysts arise from multiple mutational events of WDR35 loss, or are they clones? So are they single colors? And very neatly, what we identified was actually small isolated cysts that don't progress onto polycystic disease are pretty much always clones. So they always form like this, and they don't form anything more complex. Whereas actually in polycystic disease, where the cysts have progressed on to this large structural change where you will have multiple cysts that have multiple lumens, we found that within one face of a cyst and the adjacent face of the adjacent cyst, they would share the same color. And so this suggested to us that instead of coming from two mutational events, what actually happens is that some of these cysts form from essentially a mechanical separation of a mother cyst into two daughter cysts. And we've kind of termed that cyst fission. And we think this explains why cystic disease has a typical pattern of things like von Meyenberg, where these many small cysts sit adjacent to each other. We think that's because the parental cyst essentially gets so large, it divides and forms structural progeny. So in that case, our alpha-2 integrin inhibitor should reduce the ability of cysts to divide and form daughter cysts. And so we went back to our tissue where we saw no change in the size of the cyst and looked at cyst number, And that's exactly what we see. So we see that actually when we treat with an alpha-2 beta-1 inhibitor, we lose the amount of phosphomycin light chain within those cysts. And we also significantly reduce the number of cysts per millimeter of liver. So we think what's happening here is that this procystic ECM, which is fibronectin rich and driven by TGF-beta, is perceived by these cyst-specific integrins. And those integrins are driving a cell-autonomous change in the cytoskeleton, and that is causing those large cysts to then undergo structural division into what we see histologically as polycystic disease. So what can we conclude, I guess? So making and maintaining ducts is a complex business. You need multiple signals to be integrated together. And it's not enough just to have diffusible signals. You also need to understand tissue mechanics in order to form these very complex structural parts of the liver. We think in development, we found a way in which spatial information is integrated with tissue mechanics to regulate elongation and formation of ducts. And we think that cilia are required to maintain that structure. And when you lose cilia, essentially what we see in polycystic liver disease is a change in duct width that becomes pathological and then undergoes this cyst fission process in order to form what we ultimately see in the patient. So I appreciate that's a bit of a kind of tour de force and probably quite different to what you've heard before. But I hope I've kind of shown you some of the exciting developmental biology that we do and how we can use some of those principles to really understand both kind of like adult disease and how those will be potentially applied to cancer. So within the lab, that's my group at the top. So Scott did a lot of the polycystic liver disease work, and Michaela Raab worked on the liver development things. And I'm very happy to take questions. So thank you. Of course. So I think it's that has been one of the cornerstones of our work, right? So understanding how those pathways in development regulate regeneration and also then used by cancer. So we've previously seen that the Notch pathway particularly is very highly upregulated in cholangiocarcinoma and regulates, we think, cellular proliferation in the context of cancer. We are less convinced that how should I say this? So no, I'll change my thoughts slightly. So Notch definitely there. The Wnt pathway is interesting. So we definitely have beta-catenin activators, so canonical Wnt signaling, which we don't think is activated in regeneration. But what is strange is the work I showed you on planar cell polarity. Cholangiocarcinoma is lots of new ducts forming, but without any control. And so we see a lot of the same planar cell polarity signals expressed in cholangiocarcinoma as we see in developmental biology and in regeneration. And so there seems to be this kind of tube forming program or duct forming program that is turned on even in the cancer context. And we think that's quite important because we don't see that same program turned on in health. So it gets turned off if you're a normal bile duct, that gets turned off. So this gives us something that we can potentially target to stop cholangiocarcinoma growth independent of what's causing the cholangiocarcinoma, so the mutational profile. So a very, very interesting question. So one of the things that we definitely see in our mouse mutant where we delete planar cell polarity is that you still seem to form the very large ducts. So particularly the ducts going into the hilar and the first and second order ducts. You then lose connectivity in the smaller ducts. So I think, and we've not formally proven this, but I think those small ducts tend to be more sensitive to loss of polarity. And I suspect that what we're looking at in cancer reflects something where I've said, planar cell polarity is important. I think that's more intrahepatic. I think if we look at perihilar cancers and distal cancers, we probably won't see the same pattern of planar cell polarity. Yeah, so again, a great question. So patients with WDR35 mutations have syndromic disease. So they have polycystic liver disease, but they also have a number of other clinical derangements. So those patients definitely get polycystic disease. So in normal polycystic disease, so polycystic disease driven by things like mutations in PKD1 and 2, we think that the levels of WDR35 are probably the same. What we think happens there is that you can no longer sense your extracellular microenvironment through PC1, so the receptor on the cilia, and that causes the same phenotype. And what we've now got is some nice data from patients with WDR35 mutations, PRKCSH mutations, PKD1 and 2 mutations, and SEX63. And all of those patients get polycystic liver disease for very different reasons, but all of those patients have exactly the same proteins, so TGF-beta receptor 2, SMADs, integrin alpha 2, expressed on the biliary epithelium. And so we think that this is a common cellular response to loss of signaling through the cilia, but it doesn't necessarily matter how you lose that signaling through the cilia. That can be structurally, like with WDR35, or that can be through sensation by the cilia. So no, we actually really expected to see YAP, and we didn't see it at all. We were almost a little surprised not to see it. What we tend to see, so TGF-beta, as I showed the data, is quite heavily enriched. A lot of the integrins, and then strangely quite a lot of pro-inflammatory cytokines. So we are currently looking at how the cystic epithelium regulates a kind of potentially more inflammatory situation around itself compared to the normal dose. But no, that's a really nice question, because I think we imagine that things like YAP being mechanosensitive would change, but that wasn't the case. I mean, it's a big question. So I think it would depend very much on the type of disease, the type of mutation perhaps that the patient has. So if you've got a disease, a mutation in something like WDR35, where you have a defect in loading cargo into your cilia, then potentially, yes, you could overcome that somehow. If you've got a mutation in something like PKD1 and 2, which is put into the cilia normally, but then doesn't signal, then trying to figure out how you would, because the cilia is there, it just doesn't work. Trying to figure out how you would target that therapeutically, I think would be more challenging. But again, this is perhaps where thinking about the broad cellular processes, so things like integrins or TGF-betas, is beneficial to trying to reciliate a cell. So instead of treating the cause of the disease, you're treating the symptomatic effects of ciliary loss. That's again kind of something that we've thought of but not looked at. So I'll address the idea of duct length first. So we don't think the cilia is important necessarily for duct length. We think the cilia is presented into the lumen of the duct very late, so just before birth. And so by that point, if everything's patterned normally, you've already formed a biliary tree. So we think the cilia presenting into the lumen is part of locking your duct into its kind of phenotype. In regeneration, we've not looked in partial hepatectomy, which is a cool idea, because essentially we've only looked at inflammatory regenerative models. We've not looked at anything that would be essentially kind of sterile. In duct regeneration, generally we don't see a loss of cilia, but I want to caveat that very heavily with the idea that we're not actually very good in the mouse, at least, at looking at regionalization of regeneration. So when we just take a cross-section look at cilia, it might be that we're missing a small population of cells that's regenerating, a duct from a leading edge or something. So whether those cells lose cilia, I don't know. Yeah, we don't. We've not knocked out IFT88. IFT88. What we do know is that if we delete PKD1 and PKD2, then those mice will develop cysts. So it is not simply that this is WDR35 phenotype. Essentially, if you take out other cilia components, you will. So there was a paper a couple of years ago showing that KIF3A, which is another kind of cilia component, deletion of that in the biliary epithelium will generate kind of cystic looking structures. Interestingly, with that though, you don't need to you do need to injure the livers in order to get cysts. So the WDR35 we use, you don't. Whether or not that's because WDR35 is mutated, well, when you have WDR35 mutation, you get fibrocystic disease. So there is an inflammatory component to the cyst. Whether that's directly because of WDR35 loss, I don't know. And whether KIF3A doesn't quite give you the same inflammatory or fibroinflammatory microenvironment, I'm not sure.
Okay, thank you. So I think that there are no more questions. So I think that it's time to finish. So thank you very much, Luke, for your talk and for being here today. And thank you all for attending this seminar today. Thank you.
Thank you so much. Thank you for having me again.
Buenas tardes.